Okay, this mini lecture is designed to help you understand genome replication strategies. So really what we're talking about is how can the virus make more of its own genetic material. So remember that a virus has either DNA or RNA as the genetic material. And this genetic material has to be replicated to make more virus. Okay. So this is the same idea as if your cells are going to divide through cell cycle mitosis. You have to replicate your chromosomes so that you have more genetic material than to split to the different cells. So just generally think of it like this. You've got a virus and it has some kind of genetic material. We're not going to worry if it's DNA or RNA. And when it gets into the cell, it needs to copy that genetic material so that it can be, repackaged. Sorry, these are ugly virions. Right? And so you can go from 1 to thousands of viruses. So right now all we're focusing in is how you're going to copy the DNA or RNA genetic material. So we're going to talk about DNA viruses first. And what I want you to remember is that in DNA replication, there's a few important steps. First of all, you have to have proteins that recognize the ORI, or origin of replication, to initiate DNA replication. Okay. So at these ORIs, whoa, that's not so good. These ORIs are actually specific sequences that in our cells we have proteins that recognize the origin, recruit helicase, remember helicase unwinds the DNA, right? And then we get bidirectional synthesis. And remember that DNA polymerase can only synthesize 5' to 3'. So we're not going to go into the details. Remember leading and lagging strands and Okazaki fragments and all of that. But I want you to understand that DNA replication in viruses has to have some of the same steps. You have to be able to recognize where to start. You have to be able to recruit all of those replication enzymes. Okay. DNA polymerase requires, let me redraw it here, an RNA primer. What it really requires is a 3' hydroxyl group. So DNA polymerase cannot just start copying on its own like RNA polymerases can. So in eukaryotic cells, our cells make an RNA primer using primase. DNA polymerase comes on and replicates, and then eventually the primers are destroyed. Okay. The problem with primers is the end replication problem. So here, where there was a primer, when that RNA is removed, you've got an end of DNA that you cannot copy. Eukaryotic cells deal with this by putting telomeres, so lengths on the ends of our chromosomes that are non-gene coding and can be lost. And you see each round of DNA replication, you lose 50 to 200 base pairs. Okay. That's fine. We have telomeres they're long, and each time they're shortened a little bit. But in eukaryotic cells, as the telomeres get to a certain length and they get too short, the cell will undergo apoptosis, which means it's programmed to self-destruct. Because what's going to happen is if you kept doing replication, you would start to damage or lose important genetic information. Okay, so viruses need to deal with this end replication problem because they want to live in quotes, you can't see me, forever, right? So they don't want to eventually lose genetic information. The other thing is that if a virus is dependent on the host cell with enzymes like DNA polymerase and helicase, then the virus has to be able to program the cell to go into S phase. So remember your cell cycle, you had G1, S, G2, and mitosis. So S phase is where all the DNA replication enzymes are being expressed and active. So a virus has to do a couple different things in order to replicate its DNA genome. All right, so there are 3 classes of DNA viruses, and I'm just going to make a note because I realized this is a bad slide. Please, please, please put Baltimore classifications in Roman numerals. Okay, and now I understand why some of you write them as regular numbers because I have a crappy slide in here. Okay, so we're going to have class 1, double-stranded DNA, class 2, viruses with a single-stranded DNA genome, and class 7, viruses with a double-stranded DNA genome that use reverse transcription. Okay, so we're going to look at some of the strategies for these different classes of viruses. So this is class 1. These are double-stranded DNA viruses. And the easiest way to avoid the end replication issue, and that's really what I'm focusing on here, because what you figure out for each of your individual viruses is what kind of proteins do they encode. So do they encode their own helicase? Do they encode their own DNA polymerase? How is the origin of replication recognized? Those are all proteins encoded by the virus. What I want to focus on here is how that double-stranded DNA is copied or replicated. And the main issue is to avoid the end replication issue, right, which is the loss of DNA. And the easiest way to do that is to circularize the genome. And so this is just 2 examples. SV40 has a circular double-stranded DNA genome. So it encodes, for example, the large T antigen, which recognizes the origin of replication and recruits replication enzymes, and then replication just happens, copying both strands, and you end up with 2 double-stranded circular genomes that could then eventually be encapsulated. Okay. Another option, herpes simplex virus over here, has a linear genome. So what it does is it circularizes. And a lot of viruses have what we call, oh my gosh, my brain, ITR. Here we go, inverted terminal repeats. So these are like complementary base pairs that can come together and bring that, in this case, linear genome into a circle. And then you just do the same kind of thing. You get replication complexes on, and in this case, herpes does it kind of different. It copies around the circle, and then it just keeps copying both strands. Every DNA polymerase we've ever seen can only copy 5' to 3', so you always have to deal with leading and lagging strands. But as you go around the circle, what this virus does is just continues to replicate. It's called rolling circle. And then it cleaves the individual genomes, so now they're back to linear, and packages up them in a virion. Some viruses take a different option. Here's another double-stranded DNA virus. And again, what it has is these inverted terminal repeats on the end, so that the DNA polymerase can use a self-primer. So instead of having to use an RNA primer that will eventually be destroyed, it uses a piece of DNA. So that actually cuts or nicks the DNA, makes this 3' hydroxyl end, and now DNA polymerase can just go ahead and start copying, and then pox viruses use this interesting method of folding back these inverted terminal repeats and then allowing it to copy the other strand, and what you eventually get is more genomic DNA. Right? So remember, the whole point of genome replication is to make more of whatever is packaged in that virus. The example I have of you for a single-stranded DNA virus, so this is a class 2, class 2 virus. Similar idea, it has a free 3' hydroxyl here already because it's single-stranded, and the DNA polymerase just comes along and starts copying, and then it also has inverted repeats. And so the way the single-stranded DNA genome is packaged is it, again, makes these things called concatenamers, which just means you repeat, you repeat, you repeat, you repeat, but it's all hooked together like a link of sausages. And then as the capsids form, you just package one strand of that, and then you package the next strand, and that is how you copy the genome and then get it into the capsid. So, what do I want you to understand from this? The take home is that DNA replication for viruses, let's put viral DNA replication, is very similar to cell DNA replication. And so the virus has to encode proteins that might allow for the ORI recognition. Some viruses encode their own DNA polymerase, or they have to be able to induce the cell into S phase so that the host cell DNA polymerase is produced. And it has to deal somehow with primers, okay? To avoid the end replication issue. Okay. So these are unique to each virus, depending on what they encode, and how they deal with this end replication issue is what I was just showing you in the last slides. Being circular, circularizing, having inverted terminal repeats, somehow avoiding the use of an RNA primer like our cells do. We can't forget the class 7 weird double-stranded DNA virus that uses reverse transcription. And hepatitis B virus is just really cool but strange because it has a partial double-stranded DNA genome. And what it does, in fact, is, and look at this red right here, it actually makes an RNA molecule that is encapsulated. Okay, so it's green. Its RNA is encapsulated. And then it uses reverse transcription. Now remember, transcription is DNA to RNA, so reverse transcription, oh sorry, I'm in the kitchen. Reverse transcription means RNA to DNA. So this is just a weird virus. It packages RNA. It uses reverse transcription to eventually make this double-stranded DNA genome. Strange. But again, the purpose is make more of this genome to package into virions. Okay, RNA viruses, so viruses that have RNA as its genetic material. We have 4 classes here. We have single-stranded RNA, we have double-stranded RNA, and then we've got an RNA that uses reverse transcription. So let's look at how those replicate their RNA. What I want you to realize is for all of these guys, there is no DNA involved. In fact, the enzyme for replication is RDRP, and RDRP means it's an RNA-dependent RNA polymerase, which if you break it down means it makes RNA, that's what an RNA polymerase does, but it copies it from RNA. So RNA Pol2 that you learned about for transcription in eukaryotic cells, RNA Pol2 is a DNA-dependent RNA polymerase. We just never use all that information. But RDRPs means they copy RNA to more RNA. All right, so whether you are a, I had to look at my classes, whether you are a class 5 or a class 4, okay, so that means your genome is either positive stranded or negative stranded. Really doesn't matter, because what you have to do is you have to copy your genome to the opposite sense and then copy it back. So if you're a positive, you're going to copy to a negative, and then you're going to copy back to a positive. So this is genome to more genome. And that works the same. If you're a negative, you're going to copy it to a positive and copy it back to a negative. And the key here is that the RNA-dependent RNA polymerases do all of the RNA replication. Okay. So keep it really simple. Nothing too exciting here. We're just copying RNA to RNA, and what's important is you have the right sense, either positive sense or negative sense, to be packaged. Remember that positive sense means the same as mRNA. So a positive strand can be translated, and we'll talk about that in the next lecture. The negative strand cannot. Okay. So here is the mRNA for negative stranded viruses. Here is the positive strand that can be, did I say negative strand? Positive strand, sorry, that can be translated for positive strand viruses. All right, of course we get some weird ones, double-stranded RNA viruses, and we talked about in class how double-stranded RNA can trigger an interferon response and basically trigger your immune system to kill that virus-infected cell. So, all RNA viruses have to deal with this issue. These guys deal with it by making replication happen within membrane vesicles. So they rearrange the membranes, the ER and stuff inside the host cell to kind of hide this double-stranded RNA away. Double-stranded RNA viruses like rotavirus and the retrovirus family, they do all their double-stranded RNA synthesis inside the capsid. Okay. So what's really interesting is here's the virus, okay, comes into the cell. It's got double-stranded RNA in here. dsRNA inside. And it has an RDRP. And what that RDRP does is it makes positive stranded RNA. Some of those are used for translation. We'll talk about that next lecture. Some are packaged into the capsid, along with, oh, I hate when it does that, along with, we'll put and RDRP. And then inside the capsid, the RDRP copies, ooh, I don't know what I just wrote, copies, sorry, the plus to a minus, but they stay together to make double-stranded RNA. So it's not plus to minus, minus to plus, back and forth. It just feeds in a positive strand, and then within that capsid, copies and makes the double strand, and they stay together, just like double-stranded DNA, inside the particle. We do not find negative-stranded RNA anywhere outside of the virus particle. So that's how it that is how it copies its RNA genome. All right, one more. Retroviruses, which are our class 6, like HIV. So the genome inside the capsid is positive sense RNA. And so this virus is going to take the positive stranded, single-stranded RNA, and it's going to copy it to single-stranded DNA, which is then going to be copied to double-stranded DNA. And this is done by that enzyme reverse transcriptase. Okay. What happens, which is unique with retroviruses, is this double-stranded DNA can integrate into the host chromosome. So actually, in order to make more genome, it comes along with the host RNA polymerase. So it's basically transcription. So here you can see DNA-dependent RNA polymerase makes more viral genome, which then eventually gets packaged. Okay. So retroviruses are the weird ones. They go through a DNA intermediate, and then they eventually use the host proteins to make more positive-stranded, single-stranded RNA that's packaged into the new retroviruses. So what's the take-home for RNA virus genome replication? The take-home is that RNA viruses either use RDRPs or RT, reverse transcription. Okay. These are viral proteins, so the host does not encode either an RNA-dependent RNA polymerase or a reverse transcriptase enzyme. Okay. Except for RT, there's no DNA intermediate, so you're just going RNA to RNA to RNA that eventually gets packaged. Yes, double-stranded RNA is an issue, and so different viruses have adopted different strategies to kind of hide their double-stranded RNA away so that the interferon response isn't triggered. So, summary. If you look at a virus life cycle or replication cycle, you see everything, right? You see entry and you see exit, and you see a lot of times they focus on gene expression, which is in our next lecture. But what we're talking about right here is replicating that genome so that it can get packaged. So the whole point of genome replication is to make more genomes to be packaged. Okay, whether that's an RNA genome, or in this case, I knew I had those things, a DNA genome, doesn't matter, you're making more of what was started with in that original virus. Okay, I hope that clears up genome replication a bit. The next lecture will be on gene expression, which is transcription and translation. All right, thanks.